Welcome back, folks, to another mini lecture. This particular mini lecture focuses on eukaryotic gene regulation. In particular, we're going to focus on how messenger RNA can be regulated to control the production of products. So the example that we have chosen is the case of iron regulation by cells. So iron is very important to all living cells. Too much and it becomes poisonous. Not enough, and it doesn't allow the regular metabolism of cells to ensue. So the best thing to do is first understand the general pathway of the central dogma, where we have in the nucleus, let's just draw a nucleus here, we have DNA, and the DNA is first transcribed to pre-messenger RNA. Okay, and that is from the promoter to the terminator of the gene. And therefore, you get your pre-messenger RNA. Then the pre-messenger RNA can be processed to form messenger RNA. Subsequently, the messenger RNA is then exported out of the nuclear pore into the cytosol. And in the cytosol, the messenger RNA is destined for the ribosome, where translation takes place. What we're going to focus on today is a particular step in this pathway that can regulate the behavior of the end product, which are some proteins produced by the ribosomes. Two proteins in particular. So we're going to intercept the system right here at this point. So this is a translational control point involving messenger RNA. So our focus will be at this location in this pathway of gene expression. So let's set the stage for this particular example. So cells need iron. So here's a cell. Here's the nucleus, and the cell needs iron. And iron arrives through the food supply and it's carried in the blood by a protein. And this protein has two sites. The first site of this protein is where the iron is carried. Iron is a metal, so it carries this very positively charged entity. And this protein here is called transferrin. Transferrin. Very important that we know that. It's easy to remember the name of the protein because what does the protein do? It transfers iron from the gut to the cells. Okay, so transferrin. So that is this protein here. The second thing to notice is that the protein has a docking site on its structure. And this docking site is compatible with a cell surface receptor for transferrin. And that's obviously called the transferrin receptor. So that's the transferrin receptor. The docking of the transferrin with its receptor results in a change in the cell's behavior such that, if we can draw this over here, the complex is internalized into the cell by the formation of an endocytic vesicle. So this is phagocytosis, right? So the protein ends up bound to its receptor like this, and then we have the iron sitting inside this invaginated pocket on the surface of the cell. Now what happens next is crucial. The internalized vesicle and then moves to the center of the cell, such like this, Remember, this is a membrane enclosed structure, and the protein is now within, the transferrin is now within the cell, and it's sitting obviously on its receptor like this. So we simply move from here to there. So the system travels in that direction. Once the vesicle has been internalized, the iron is released. And the iron, in a way that's not important right now, is then moved into the cytosol. So now we have iron inside the cytosol. So this is where things become really interesting. If this particular cell is deficient in iron, so it has low levels of iron, then one thing happens. So when iron levels are low, the iron is then used by the proteins which need them. Right? So the iron molecules are picked up by various proteins in the cytosol of this cell, and indeed even in the nucleus. So that's one thing. But when iron levels are too high, the importation of iron continues because iron is such an important commodity. But this time, the iron cannot be allowed to remain in surplus in the cytosol because it's toxic. So what does the cell do? It puts it inside a vault. The iron is stored inside a vault. What is this vault made of? It's made of proteins. And the proteins that the vault is made of, so let's put this here, vault. And the iron is then transferred into the vault. And what is this vault made of? It's made of a collection of proteins which form a hollow sphere in the cytosol. And the iron is transported into that sphere, and in essence, it's removed from circulation, right? And the name of that protein complex, the vault, is again very similar to the name transferrin. It's called ferritin. Easy to remember. Ferret means iron, in means inside the vault. There you go. So that will be your iron-containing vault. So the iron is then stored in there until it's needed in the future at some time. So there's the pathway. So the question is, how are various decisions made? How are various decisions made within the cell as to what to do with the iron? How is the level of iron detected? How is iron imported into the cell and then sent down these alternative pathways? And that in itself is amazing. And that's what we're going to learn with this particular example. In order to fully understand this very simple circuit, we need to follow the fate of two proteins that are produced by a cell. So the two proteins, let's put them in black, the two proteins will be the transferrin receptor and the vaults, which are ferritin. Because these two products are proteins, which are made by the cell that we're going to be looking at now, they have to be made by ribosomes. And both of these are made by ribosomes, which are located in the cytosol of the cell. So let's draw the cell and put that information together for ourselves. So here's the surface of the cell with the plasma membrane. And then we're going to draw in a couple of ribosomes. Let's put the ribosomes in blue. So here's a ribosome. Here's another ribosome. Let's assume that the top ribosome receives from the DNA a messenger RNA. Let's put the messenger RNA in red. So we have messenger RNA for the transferrin receptor. Okay, so this messenger RNA is for this receptor here. So under normal circumstances, the messenger RNA will be released by the nucleus on the left-hand side somewhere. So we can put the nucleus in for the sake of completing this figure. So the nucleus will be here somewhere. And inside the nucleus, we have the genes on the DNA for producing these two proteins. So the messenger RNA is then translated into a protein. Let's put the protein in orange. And the protein then goes to work by sitting in the plasma membrane. So that's going to be your transferrin receptor. TR stands for transferrin receptor. Okay, so that's how these products are made in the normal course of what we understand is the central dogma for protein production. The same thing happens with the gene for ferritin. The gene for ferritin, let's put it in a slightly different color because there are differences between the way that the two messenger RNAs are handled within the cell. Okay, so let's use a different color here. Let's use a color that we haven't really used much. Let's go for green. Okay, so the messenger RNA for ferritin is also released from the nuclear pore when it's mature, and then it makes its way to the ribosome. When it gets to the ribosome, it's translated into this polypeptide, and that polypeptide then goes on to make the vaults, the ferritin vaults, right? So here we are. These structures are the ferritin vaults. And they come together automatically once they're assembled, right? So that will be your ferritin. Now, depending on the needs of the cell, the number of receptors can be regulated. So the cell can make a full complement of receptors, or it can reduce that number by simply altering one of these steps in the pathway for their production. Likewise for ferritin. And the most suitable place that nature has chosen to regulate this behavior is at the messenger RNA level. So regulation takes place, in this case, at this step and this step. The two systems use the same general principle of interfering with the messenger RNA. So if the messenger RNA is interfered with and is prevented from getting to the ribosome, then naturally no polypeptide will be made for either of these two proteins. And that's the key. The key is now, how does this circuit work? And it's very elegant and it's straightforward. We just need to look at the structure of a couple of other proteins that make the decisions for the cell. So we're going to look at the example of transferrin second. We're going to look at the example of ferritin first. So on the next slide, we're going to deal with ferritin. Let's draw a typical RNA molecule here and divide it into three zones. We have the 5' untranslated region. The region that's not translated into protein. Then we have the coding region, the part that is converted into polypeptide. And then at the end, we have a zone called the 3' untranslated region. And if you recall, this is eukaryotic messenger RNA, so you should have a poly A tail on one end and a cap on the other end. And during regular translation, the ribosome will bind to the cap and then travel down until it finds the first start codon, which should be here. And that will code for methionine, AUG. Okay, and then of course there should be a stop codon at the end of the coding region. The most important part of this messenger RNA when it comes to this particular protein, ferritin, is the 5' untranslated region. So that's where the magic decisions are made. The decisions are made by a particular zone on the untranslated region called the IRE. The IRE is a small region of nucleotides that bind to a protein, and that protein is key in this decision making. So even though this is RNA and it's single-stranded, it can still carry information in its nucleotides. So that region there is called the ion response element. So it's not just DNA that can have elements, RNA can have elements too. So the most important player when it comes to this system here will be a protein known as ion regulatory protein, IRP. And let's give the IRP a shape. There we go. Let's make it look like a pizza wedge. And it has two binding sites. It has a binding site there, and then it has a allosteric binding site there. So the number one site is for the IRE. So any messenger RNA that's in the cytosol in the presence of this protein will then bind the protein to the IRE. Now this protein can detect the amount of iron within the cytosol. So it acts as a switch. So the two conformations that we need to worry ourselves with is number one, when iron levels are low, what happens? And then alternatively, we need to consider what happens when iron levels are high. So let's take the first scenario, right? Let's change the color so we can follow that. When iron levels are low, let's use purple. When iron levels are low, there's no need to translate this messenger RNA. Why? There's no need to store extra iron because there isn't any. Iron levels are low, and any iron coming into the cell will be used by the biochemical machinery. So you don't need to make these vaults. So how is the choice made by the cell? The choice is made by the IRP. When there's no iron, nothing binds to the allosteric site. Nothing binds there. So the protein has a natural affinity, a liking for the IRE. And that's exactly what happens, right? So the protein will be found, in this case, attached to the IRE. Now, when the protein is sitting on the IRE, it makes it impossible for the ribosome to bind and then move along past that particular protein. Therefore, translation is prevented, and therefore this messenger RNA does not produce any protein. Right? So no ferritin is produced. That makes sense because there's not enough iron for you to store within the ferritin complex. Let's look at the alternative scenario. When iron levels are high inside the cell, let's use a different color to follow that. When iron levels are high, what happens? Well, iron diffuses throughout the cell and it binds to the binding site, the allosteric binding site on the surface of IRP. That causes a conformational change at the active site of the protein. Therefore, it can no longer bind to the IRE. So in this case, let's use the eraser to get rid of what we don't need. So in this case, we end up with this situation, okay? And because IRP is no longer bound to the IRE, the ribosome has free rein in now copying our messenger RNA into a polypeptide, and therefore ferritin will be produced. A very simple and elegant switch that all relies on sensing the amount of iron within the cytoplasm of this particular cell. Straightforward and simple, right? Block the ribosome from attaching properly and translating the messenger RNA. So the messenger RNA will not be destroyed, it'll just be blocked. And it's sitting there in the cytosol for the time when it is needed. So you don't have to go all the way back into the nucleus to activate the gene for producing ferritin. Now let's turn our attention over to transferrin. Okay, so we're going to look at how transferrin is regulated. It's a very similar system, but it works in reverse. So this is the receptor that will be sitting in the plasma membrane waiting to dock with transferrin. So again, it can be divided into 3 zones. Okay, so we have the 5' untranslated region, we have the coding region, and then we have the 3' untranslated region. And it'll have a poly A tail and a cap on the front end. Okay, so in this case, nature has come up with a beautiful scenario where we have the start codon here and the stop codon there. This time, the IRE is not found at the front of the messenger RNA. No, it's the reverse. It's found at the back end of the RNA. So right there, somewhere here, we will have our IRE, the iron response element. Okay, it's sitting at the far end of the molecule. Unlike in the previous example, with that particular protein, ferritin, which is in the front end, with transferrin, it's in the back end. The same protein regulates this behavior too. So again, it's the same protein, right? So the protein that we're talking about will be this one here. So this is a commonality inside a cell that one protein can perform multiple tasks. So in this case, this protein, which we know the name of already, iron regulatory protein, it works in exactly the same way. So as we told you before, there's 2 scenarios, right? When iron levels are low, we need to predict what happens, right? So low iron levels. And we then need to predict what happens when iron levels are high. And that's it. So can we do that? Now think about the logical situation, right? When iron levels are low inside the cell, you want to capture as much iron from the outside and bring it in. So you need this receptor to go fishing for iron on the cell membrane. So let's start with that scenario first, right? So when iron levels are low, you need the product of this messenger RNA, which is transferrin, to be produced. How does that work? Well, when iron levels are low, what happens is that they do not bind to the allosteric site on the protein. And now the protein is free to drift and bind to the IRE. So that's what happens. When iron levels are low, the protein, the IRP, is found sitting right here, like that. Okay. What it does is it protects the back end of the messenger RNA from being attacked by other enzymes. Those are going to be the different enzymes to the ones that attack the poly A tail. These enzymes want to eat this part of the molecule, digest it, to break it down, right? So but the presence of the protein, the IRP, protects that end of the messenger RNA in addition to the poly A tail. So this is extra protection. That makes the messenger RNA available for longer inside the cell, and the ribosomes in this case will attach at the 5' end, and then they'll be free to translate this messenger RNA a number of times. So you're keeping the messenger RNA inside the cytosol longer, so lots of protein receptor will be made, and then it will be sent to the surface of the plasma membrane to act as a receptor for binding the transferrin chain. Okay, so that's what happens here, right? So low iron levels lead to more receptor production. So let's look at the second scenario. So in this scenario, when iron levels are high, you don't want to bring in any more iron because you have enough, okay? So what happens then is that the increased levels of iron, they drift across and then they bind to the allosteric site. That changes the configuration of the protein. Therefore, that protein no longer binds to the IRE. So let's get rid of this line. And therefore, it's not sitting here. Let's just get rid of this too for the time being. It's not sitting there on the messenger RNA. And now our enzymatic friends are free to attack the messenger RNA. And when they do that, they cut it into pieces, beginning at the 3' end. And once you start cutting it into pieces, the messenger RNA, it doesn't survive very long, and it's destroyed. It's no longer available to be translated into transferrin receptor, and therefore the number of receptors is reduced on the surface of the cell, and therefore less iron enters the cell.